The most important, definitely the most complex energy engine on our entire planet.
That's spot on. Photosynthesis isn't just some simple formula. I mean, it's the molecular bedrock for almost all life, for the air we breathe, for the food we eat. It's huge.
So our mission today in this deep dive is really to pull apart that simple concept. We're going way beyond the textbook summary.
Yeah, we're digging into the sources that detail the incredibly sophisticated machinery inside the chloroplast that actually does the work, the structures, the pigments, the reactions.
And the sources also cover these like amazing evolutionary workarounds plants have developed to keep the whole thing running smoothly. So basically you're getting the inside track today on the biological light engine that quite literally powers the world.
Okay, so let's start big picture, maybe with the chemistry first, because at its heart, it's all about energy and chemical bonds.
Good idea. So the main goal is what? Taking sunlight.
Taking that temporary solar energy and locking it away into stable, usable chemical energy. Specifically, we're talking ATP and NADPH. Those are the key energy carriers.
Right, the cell's rechargeable batteries, basically.
Pretty much. And then the plant uses those compounds to grab co-euro out of the air and, fix it, build it into actual organic stuff like glucose, sugar.
And thinking about the overall equation, it helps, I find, to see it as the mirror image of respiration, right?
Absolutely. Cellular respiration is all about breaking down glucose, oxidizing it, reacting it with oxygen, releasing the stored energy back into co-euro. It's like burning fuel.
Photosynthesis does the complete opposite.
Precisely. It's this huge act of reduction. You're taking carbon in its lowest energy state, co-euro, fully oxidized, and using light energy to force electrons back onto it, building it up into complex high-energy sugars like COROs.
So the inputs, 6 CRO, 6 waters, plus light energy.
Yields 1 glucose molecule and 6 molecules of oxygen. It's building stuff, storing energy, and changing the atmosphere all at once.
And the scale of this, I mean, to really get how powerful it is, we need to look back in time, way back. Because when this first evolved, it wasn't exactly a gentle process for the planet.
No, not at all. You're talking about the Great Oxygenation Event, the GOE. This was, maybe 2.4 billion years ago. The first organisms to figure out oxygenic photosynthesis, these ancient cyanobacteria, they didn't just change themselves. They fundamentally rewrote Earth's atmospheric chemistry.
And here's the crazy part, the paradox. These little guys started splitting water molecules, H0, to get electrons for the process.
And the waste product was oxygen.
Yes.
Oro, lots and lots of it, pouring into the oceans and eventually filling the atmosphere.
Which sounds great to us now, but back then...
It was poison. Pure poison to almost everything alive at the time. Most life was anaerobic. They couldn't handle oxygen. It was toxic.
So this metabolic breakthrough triggered what was basically life's first great mass extinction.
Arguably the biggest one ever, yeah. A planetary catastrophe driven by a new kind of metabolism.
But like you said, that catastrophe set the stage for, for us. That oxygen-rich atmosphere allowed for aerobic respiration, which is way more efficient.
Exactly. It unlocked the potential for bigger, more complex, more energetic life forms. So the very process that nearly wiped out early life is the reason we're here today. It's an incredible story of adaptation.
It really puts it in perspective. Okay, so let's zoom in now. From planetary history down to the tiny, tiny organelle where all this magic happens, the chloroplast.
Right, the machinery itself. Inside the plant cell, you find these chloroplasts, and the first thing you notice is how cleverly structured they are, perfectly set up for the two main phases of photosynthesis.
There's a real division of labor, isn't there?
Absolutely critical. You've got the outer and inner membranes, kind of like the building walls, controlling what goes in and out, but inside that, you have the stroma.
The stroma is like the factory floor fluid.
Good analogy. It's a dense fluid, like the cell's cytoplasm, and that's where phase 2, the Calvin cycle, happens, where the carbon actually gets fixed.
But floating within that stroma, you have these other structures.
The thylakoids.
Ah, yes, the thylakoids. These are flattened sacs, almost like poker chips. And sometimes they're stacked up into columns called grana.
And why are these membranes so important?
Because the thylakoid membranes are where the action starts. They contain all the machinery for phase 1, the light reactions, all the proteins, the pigments, the electron carriers, the solar panels.
Okay, and inside the thylakoid sac itself, that space.
That's the thylakoid lumen. It's physically separate from the stroma, and that separation is absolutely key for making energy. It's like a tiny enclosed reservoir.
A reservoir for?
Protons, hydrogen ions, Hey. During the light reactions, protons get pumped into this tiny lumen space, creating a really steep concentration gradient. And that gradient is the direct power source for making ATP. Got it. So structure dictates function perfectly. Now, how did those thylakoid membranes actually grab the sunlight, the pigments, right?
Exactly, the pigments. Why are leaves green? Because they don't absorb green light well, they reflect it. They're busy absorbing other colors.
Primarily chlorophyll, that's the main player.
That's the workhorse. It's tuned to absorb light most strongly in the blue-violet and the red parts of the spectrum. That's where it gets the energy to kick things off.
But there are others too, like chlorophyll B.
Right, chlorophyll B is an accessory pigment. It absorbs slightly different wavelengths, sort of broadening the antenna's reach, and then it passes that captured energy over to chlorophyll, makes the whole system more efficient across more of the light spectrum.
And then you have the carotenoids, the pigments that give us fall colors, the yellows and oranges. But they do more than just look pretty before they break
Oh,
down.
much more. They're crucial all year round, actually. Their main job is photoprotection. They act like antioxidants.
Protecting from what? Too much sun?
Exactly. Sunlight, especially bright sunlight, can actually overload the system. The chlorophylls absorb more energy than the reactions can safely handle. This excess energy can create damaging molecules called reactive oxygen species, or ROS.
Nasty stuff.
Very. So the carotenoids step in, absorb that dangerous excess energy, and safely release it as heat. They prevent the delicate photosynthetic machinery from basically frying itself, sunscreen for the chloroplast.
Wow. Okay, so this whole setup, the double membrane, the internal DNA, which we haven't mentioned yet, the thylakoid stacks, the specific pigments, it all points to this really mind-blowing origin story, doesn't it? Endosymbiosis.
It's one of the most elegant theories in biology, and the evidence is just overwhelming. The basic idea is that way back, an ancestral eukaryotic cell, something with a nucleus, swallowed a free-living cyanobacterium, one of those oxygen-making bacteria we talked about earlier.
But instead of digesting it,
It kept it, formed a permanent partnership. The bacterium continued to photosynthesize inside the host cell. That's primary endosymbiosis.
and we can still see the fingerprints of that event today in the chloroplast structure.
Absolutely. First, that double membrane. The inner one was the original bacterium's own cell membrane. The outer one came from the host cell's membrane when it engulfed the bacterium. 2 layers.
And the DNA.
The chloroplast still has its own DNA. It's circular DNA, just like in bacteria, and it uses 70S ribosomes, again, the bacterial type, not the eukaryotic type found elsewhere in the cell. It's like a little bacterial remnant living inside.
Plus, it even reproduces like a bacterium.
Right. Chloroplasts divide by binary fission. They just pinch in half. They don't go through the complex mitosis the rest of the cell uses.
And over time, the integration became complete.
Yeah, the evidence shows a lot of the original bacterial genes actually migrated to the host cell's nucleus. This cemented the relationship. The chloroplast can't survive on its own anymore. It depends on the host nucleus for many essential proteins. It's fully integrated. And remember, chloroplasts are just one type of plastid. This whole family of organelles came from that one ancient event.
It's literally a captured power plant. Incredible. Okay, that sets the stage beautifully. We've got the structure. Now, how does it actually work? How is that light turned into energy? Let's dive into phase 1, the light reactions.
All right, the light reactions. This all happens on and across those thylakoid membranes we talked about. This is the energy generation step. The goal, convert light energy into those 2 chemical energy carriers, ATP and NADPH.
And the main pathway for this is the non-cyclic electron flow, sometimes called the Z scheme.
That's the one. It's called the Z scheme because if you plot the energy level of the electrons as they move through the system, it looks kind of like a sideways letter Z. This pathway gives us ATP, NADPH, and crucially, all the oxygen in our atmosphere.
Okay, walk us through it. Where does it start?
It kicks off at photosystem 2, PSII. Think of it as a complex of proteins and pigments. At its heart is a special chlorophyll pair called P680. Light energy absorbed by antenna pigments gets funneled to P680.
And that energy does what to P680?
It excites an electron in P680 to a really high energy level. This electron gets ejected immediately and passed down an electron transport chain. But now P680 is missing an electron. It's unstable, highly oxidizing. It needs a replacement, like, right now.
And that's where water comes in.
Bingo. Attached to PSII is the oxygen evolving complex. This is where the magic happens. It grabs 2 water molecules and performs photolysis using light energy indirectly to split them apart.
Into what?
into 4 protons, 4 electrons, and 1 molecule of diatomic oxygen. Those electrons are immediately used to replace the ones lost by P680. And the O, that's waste, released into the atmosphere. That's the air we breathe.
Mind-blowing every time I hear it. Okay, so PSII has its electron back. What happens to the high-energy electron that got kicked out?
That electron gets passed along a chain, first to a mobile carrier called plastoquinone, PQ. Think of PQ as a little ferry boat in the membrane. It picks up the electron from PSII and carries it over to the next big complex, the cytochrome B or Faf complex. Now, this complex is really interesting. It doesn't use light directly, but it uses the energy lost by the electron as it passes through.
Lost how?
As the electron moves through the various components within the Cyt or BFF complex, it steps down in energy level. That released energy is harnessed by the complex to do work.
And that work is pumping protons.
Exactly. It actively pumps those He ions, the ones released from splitting water, plus others from the stroma into that tiny thylakoid lumen space. This starts building up that proton gradient we mentioned earlier, really concentrating them inside the lumen.
So the electron flow directly powers the proton pump.
Mm-hmm.
Got it. What happens to the electron after it leaves CBF? It's lower energy now.
It is. It gets passed to another mobile carrier, a small protein called plastocyanin, PC. PC then delivers this lower energy electron to the second photosystem in the chain. Photosystem I, PSI, which has a reaction center called P700.
Why another photosystem?
Because the electron has lost too much energy going through Cyt BF, it doesn't have enough juice left to do the final step. So PSI acts like another solar panel. It absorbs another photon of light, using that energy to re-excite the electron back up to a very high energy level, again, even higher than it was at PSII initially.
Okay, re-energized electron at PSI. Where does it go next?
It's passed to another carrier called ferredoxin, FD, which sits on the stroma side of the membrane. Ferredoxin is the key link to the final enzyme in this non-cyclic path, ferredoxin and adiporeductase, or FNR.
FNR. And its job is?
Its job is to take 2 of these high-energy electrons from ferredoxin, grab a proton from the stroma, and use them to reduce NADP, which is floating around in the stroma, into NADPH. That's our second energy currency molecule, high-energy electron carrier.
Okay, so non-cyclic flow gave us NADPH, and it pumped protons into the lumen. Now, how do we get the APP?
From that proton gradient, the lumen is now packed with atom ions. It's acidic and has a positive charge compared to the stroma. They desperately want to flow back out into the stroma, down their concentration gradient.
Like water behind a dam.
Exactly. But the thylakoid membrane is impermeable to protons except in one specific place, through an amazing enzyme called ATP synthase.
And this isn't just a simple channel, right?
Not at all. It's a molecular machine, a rotary motor. As the protons flow through a channel in the base of ATP synthase, they cause part of the enzyme, the rotor, to physically spin, like a water wheel.
Seriously, it spins.
It literally spins. And this mechanical rotation causes conformational changes in the catalytic head of the enzyme, which is sticking out into the stroma. These changes force ADP and inorganic phosphate pi together, forging that high-energy bond to create ATP.
That's incredible, converting a flow of ions into mechanical rotation into chemical energy. That's chemiosmosis.
That's chemiosmosis. It's how the potential energy stored in the proton gradient gets converted into the chemical energy of ATP.
Okay, let's quickly sum up the output. For every 2 water molecules split by PSII,
You get 1 molecule of OU released, you generate 2 molecules of NADPH via FNR, and the proton pumping through Cyt B and subsequent chemiosmosis yields roughly 3 molecules of ATP.
So the ratio is about 3 ATP for every 2 NADPH. That specific ratio is important, isn't it?
Very important, because when we get to the Calvin cycle phase 2, we'll see that it actually needs more ATP than NADPH to run. If the plant only relied on this non-cyclic flow, it would run out of ATP while still having NADPH left over. It would be unbalanced.
So the plant needs a way to make just ATP sometime.
Precisely. And that's where the other pathway comes in, cyclic electron flow. It's like a little bypass loop.
How does that work?
Cyclic flow only involves photosystem I. Electrons get excited at PSI, P700, just like before. But instead of being passed to ferredoxin and then on to FNR to make NADPH,
They go somewhere else.
they get shunted back from ferredoxin to the cytochrome B of complex.
Back into the proton pump.
Exactly. So the electrons cycle back through CytBF, driving more proton pumping into the lumen. This extra proton pumping leads to more ATP synthesis via ATP synthase.
But no NADPH is made.
Correct. And no water is split, no oxygen released. It's purely for generating supplemental ATP to balance the books, ensuring the Calvin cycle always has the right ATP-NADPH ratio it needs. A very clever fine-tuning mechanism.
Okay, fantastic. We've generated our energy currency, ATP and NADPH. Now it's time to spend it. Let's move to phase 2, the Calvin cycle.
Right, the Calvin cycle. This takes place out in the stroma, that fluid-filled space surrounding the thylakoids. The whole point here is to use the ATP and NADPH we just made to take inorganic Kyoro from the atmosphere and build it into sugar.
This is the carbon fixation part. It's a cycle, meaning it regenerates its starting molecule, right? And it starts with a really, really important enzyme, maybe the most abundant enzyme on Earth.
That's the one, rubisco. Ribulose 105 bisphosphate carboxylase oxygenase, quite a mouthful. It's the star player in the first step, carbon fixation.
What does Rubisco do?
It grabs a molecule of co-urose from the air and attaches it to a 5-carbon sugar molecule that's already present in the stroma called RoBP, ribulose 1,5 bisphosphate.
Coulose plus a 5-carbon sugar.
Right.
It says 6-carbon sugar.
Exactly. It forms a very unstable 6-carbon intermediate, which immediately breaks in half to form 2 molecules of a 3-carbon compound called 3-PGA, 3-phosphoglycerate. And boom, the atmospheric carbon is now locked into an organic molecule.
Okay, step 1, fixation, carbon captured. What's next?
Step 2 is reduction. This is where we spend the energy money. The 2 molecules of 3-PGA are converted into a higher energy 3-carbon sugar called G3P, glyceraldehyde 3-phosphate.
And this conversion requires?
It requires both ATP and NADPH. First, ATP is used to add a phosphate group to each 3-PGA. Then NADPH provides the high-energy electrons to reduce that molecule, kicking out the phosphate group and forming G3P. Energy input required.
So G3P is the actual sugar product of the cycle, not glucose directly.
That's right. G3P is the key 3-carbon sugar phosphate that comes out of the reduction phase. Now, here's the crucial bit for the cycle to continue. Most of this G3P can't leave yet.
Why not? Isn't that the prize?
It is, but if all the G3P was exported, the cycle would run out of the starting molecule, RoBP. Remember, Rubisco needs RuBP to grab the next Kyoro.
Ah, so most of the G3P has to be used to remake the RuBP.
Exactly. That's the third and final phase, regeneration. For every 6 molecules of G3P produced in the reduction phase, which required 3 CO molecules to enter the cycle.
Only one G3P gets to leave.
That's the net output. One G3P molecule is exported from the chloroplast to be used by the cell to make glucose, sucrose, starch, whatever the plant needs. The other 5 G3P molecules must enter a complex series of reactions
To regenerate the initial RuBP.
Yes. Those 5 3-carbon G3Ps are rearranged and eventually converted back into 3 molecules of the 5-carbon RuBP. And this regeneration step isn't free. It costs more ATP.
So let's tally the cost. To get just one net G3P out, a 3-carbon sugar, the cycle has to turn 3 times, fixing 3 Kyaros.
Right. And that requires a total of 9 ATP and 6 NADPH.
Okay, but plants usually store energy as glucose, which is a 6-carbon sugar. So to make 1 glucose,
You need 2 net G3P molecules. That means the cycle has to turn 6 times, fixing 6 anhydrous molecules.
and the total cost for 1 glucose molecule.
A whopping 18 ATP and 12 NADPH. It's energetically very expensive, which explains why plants are so concerned about efficiency and avoiding waste.
Which brings us perfectly to the next big topic, the trade-offs. Because that superstar enzyme, Rubisco, has a dark side, a pretty major flaw, actually.
Oh yeah, it's a huge headache for plants. Remember the full name? Ribulose 155 bisphosphate carboxylase oxygenase. That oxygenase part is the problem.
Meaning it doesn't just bind TRO?
Unfortunately, no. It can also bind over-aero oxygen gas. And when it does that, instead of initiating photosynthesis, it triggers a wasteful process called photorespiration.
What happens in photorespiration?
Instead of fixing CoO onto RoBP to make 2 useful 3-PGA molecules, Rubisco binds O to RuBP. This produces only 1 molecule of 3-TGA and 1 molecule of a 2-carbon compound called 2-phosphoglycolate.
And that 2-phosphoglycolate. Bad news.
It's basically toxic waste. The plant has to spend a significant amount of energy using up ATP and involving multiple organelles just to break it down and salvage some of the carbon. Worse still, during the salvage process, it actually consumes OO and releases CoO.
The exact opposite of photosynthesis.
Exactly. It undoes the hard work of carbon fixation. It's incredibly wasteful, especially under certain conditions.
Like when? When does Rubisco tend to grab oxygen instead of CoO?
It happens more often when the concentration of ORO inside the leaf is high relative to the concentration of CoO. And when does that happen?
When it's hot and dry, the plant closes its leaf pores, the stomata, to save water.
Precisely. Stomata close, co-urose can't get in easily, so the CO level inside the leaf drops as the Calvin cycle uses it up. Meanwhile, the light reactions are still splitting water, pumping out ORO, so you get low CORO, high ORO, perfect conditions for photorespiration.
And plants that only use this standard Calvin cycle, we call them C plants, right? Things like rice, wheat, soybeans.
Yes, the vast majority of plants are C, and on a hot, sunny, dry day, they can lose a massive chunk, sometimes up to 50% of the carbon they just fixed, all because of Rubisco's mistake. It's a huge drain on their growth and productivity.
So evolution needed a workaround. Plants couldn't easily fix Rubisco itself, so they evolved ways to sort of manage the environment around it.
Exactly. Two main strategies emerged to basically force feed coir to Rubisco and keep oxygen away from it.
The first one involved seed co-row, like corn and sugarcane.
Right. Sea coos use spatial separation. They have a specialized leaf anatomy.
How does it work?
They do an initial carbon fixation step in their outer leaf cells, the mesophyll cells. But instead of using Rubisco here, they use a different enzyme called PEP carboxylase.
And P-carboxylase is better.
Much better for this initial step. It has a super high affinity for SICO, grabs it very efficiently, even at low concentrations, and crucially, it does not bind oxygen at all. No photorespiration risk here. It attaches the SICO to a 3-carbon molecule, PEP, to form a 4-carbon acid, hence the name C.
Okay, so coacease is captured efficiently as a 4-carbon acid in the outer cells. Then what?
Then this 4-carbon acid is quickly transported deeper into the leaf into specialized bundle sheath cells that surround the leaf veins. These cells are relatively impermeable to gas leak.
And inside the bundle sheath cells.
Inside those protected cells, the 4-carbon acid is broken down, releasing the CoR again. This creates a very high concentration of CoRO right where the C plant keeps its Rubisco and runs the normal Calvin cycle.
Ah, so it's like a co-pump. It concentrates CoR around Rubisco, overwhelming it so it only blinds KIOR and ignores any oxygen that might be around.
That's exactly it. Spatial separation, different steps in different places to beat photorespiration, very effective in hot, bright conditions.
Okay, that's one strategy. What's the other main one? CM plants.
Yes, crossylation acid metabolism, or CAM. Think cacti, succulents, pineapples. These plants live in extremely dry environments, so water conservation is paramount. They use temporal separation, time, not space.
How does separating by time work?
They only open their stomata at night when it's cooler and more humid, minimizing water loss.
But the light reactions only work during the day.
Right. So at night, they open their stomata, take in stomata, and fix it using that same efficient enzyme, PEP carboxylase, just like sea plants do initially. They convert the TORs into organic acids, like malic acid, and store huge amounts of these acids in their cell vacuoles overnight.
So they bank the KIOO at night. Then what happens during the day?
During the day, they slam their stomata shut tight to prevent water loss in the heat. Now, inside the closed leaf cells, they break down those stored organic acids, releasing the TORs internally.
And the timing is perfect.
Perfect. The light reactions are now running, producing ATP and NADPH. This internally released co-urose feeds directly into the Calvin cycle, using Rubisco, just like everyone else, powered by the freshly made ATP and NADPH. They run photosynthesis during the day with the doors locked, using the C-rows they collected the night before.
Temporal separation. Genius. C rates in space, C separates in time, both solving the same Rubisco oxygen problem. Incredible adaptations.
It really shows the evolutionary pressure photorespiration puts on plants. Okay, so that leads us nicely into thinking about control. We've seen the engine, the phases, the fixes. But how does the cell manage all this? It's a high-energy process. How is it regulated?
Good point. You can't just have the light reactions blazing away if the Calvin cycle isn't ready, right? You'd build up too much reactive stuff.
Exactly. There are multiple layers of control. One obvious one is light intensity itself. Light drives PSII, but it also signals to the plant how wide to open its stomata. Low light, maybe close the pores a bit, save water, no need for tons of TROs. High light, open up, let the CR flow in to match the energy production.
Makes sense. What about internal chemical signals?
A key one is redox feedback. Remember NADPH, the electron carrier produced by the light reactions. If the light reactions are outrunning the Calvin cycle, NADPH levels build up in the stroma.
Too much fuel, Not enough demand.
Right. And high concentrations of NADPH actually act as an inhibitor for some key enzymes back in the Calvin cycle. It's a direct signal. Whoa, slow down carbon fixation. We've got plenty of reducing power already. A chemical break.
Clever. And you mentioned the proton gradient earlier. Does that play a role in regulation too, the pH difference?
It absolutely does. When the light reactions are running, protons are pumped into the lumen, making it acidic. This means the stroma, where the Calvin cycle happens, becomes more alkaline. Its pH goes up, maybe to around 8.
And that pH change matters.
Critically, several key enzymes in the Calvin cycle, especially those involved in the reduction and regeneration steps, are actually activated by this alkaline pH. They work best at pH 8. When the sun goes down, proton pumping stops, the stroma pH drops back towards neutral, and those enzymes switch off. The cycle basically shuts down automatically when the light goes out.
Wow, directly linking the engine's activity to the fuel source, the light. Our sources also mentioned something called the thioredoxin system, another regulatory switch.
Yes, another light-dependent switch. Thioredoxin is a small protein that gets reduced by electrons coming from ferredoxin, part of the light reaction's output. This reduced thioredoxin then goes and activates several specific Calvin cycle enzymes by changing their chemical structure.
So again, direct proof that the light reactions are running is needed to turn on key parts of the carbon fixation process, ensures they work in sync.
Precisely. Multiple overlapping systems ensure the two phases stay coupled.
Now, zooming out a bit from just the chloroplast, it doesn't work in isolation in the plant cell, does it? It has to coordinate with the mitochondria, the cell's other power plant.
Definitely. There's a crucial partnership, especially over the 24-hour day-night cycle.
During the day.
During the day, the chloroplast is the star. It's churning out ATP and NADPH, not just for the Calvin cycle, but also providing energy and building blocks like G3P for the rest of the cell to synthesize amino acids, lipids, everything it needs to grow. Excess sugar is stored, often as starch inside the chloroplast or sucrose for transport.
And then at night? No light, so chloroplast shut down.
The mitochondria take over the main energy supply role. They perform cellular respiration, breaking down those sugars stored during the day, oxidizing them with oxygen to produce the ATP. The cell needs to stay alive, repair damage, transport substances, all the housekeeping tasks that run 204/7. It's a metabolic handover.
A continuous energy relay race. Okay, so we've gone deep into the mechanics, the evolution, the regulation. Let's talk relevance. Why do we care so much about the nitty-gritty details of, say, the Z-scheme or Rubisco kinetics? What's the so what factor here?
Well, the biggest so what is pretty fundamental. Ecological impact, we've said it, but it bears repeating. Photosynthesis produces virtually all the oxygen we breathe. It underpins almost every food chain on the planet. If photosynthesis stops, complex life stops. Period.
Can't get much more relevant than that.
And tied to that is the climate and synthetic biology angle, that inefficiency of Rubisco we discussed. The photorespiration problem, it's a massive target for research.
Because if we could make it better.
Think about it. We saw the energy cost 18 ATP, 12 NADPH per glucose, and that's before photorespiration losses. If you could engineer a Rubisco that's faster, or even better, one that only binds Kyoto and never oxygen.
Crop yields could skyrocket.
Dramatically, especially for seed crops like rice and wheat in hotter climates where photorespiration is worst. It could have huge implications for global food security. Plus, more efficient CoOs captured by crops could, in theory, help mitigate rising atmospheric CO levels. It's a major goal in agricultural biotechnology.
Okay, food and climate. What about medical applications? Seems a bit removed, maybe.
Not as much as you might think. Remember those reactive oxygen species, ROS, that carotenoids protect against? The ones caused by excess light energy?
Yeah, the damaging byproducts.
Well, if you can control the production of ROS and target it specifically to unwanted cells, you can use it as a weapon.
Ow.
That's the basis of photodynamic therapy, PDT, for some cancers and infections. You introduce a light-sensitive drug, a photosensitizer, that accumulates in the target cells, like tumor cells. Then you shine a specific wavelength of light on that area. The drug absorbs the light and generates a burst of toxic ROS, killing the targeted cells. It's essentially hijacking the plant's light stress chemistry for medical purposes, a very targeted way to destroy cells.
Wow, okay, that's a direct link. And there was one more connection related to the endosymbiotic origin, the plastids.
Yes, the apicoplast. Remember, chloroplasts are part of the plastid family, all tracing back to that engulfed cyanobacterium. Well, some nasty parasites, including Plasmodium falciparum, the parasite that causes malaria, have retained a non-photosynthetic plastid called the apicoplast.
Still derived from that ancient symbiosis, but not doing photosynthesis anymore.
Exactly. But it carries out other essential metabolic functions that the parasite needs to survive, using pathways that are distinct from human metabolic pathways because of their bacterial origin.
Making it a perfect drug target.
Precisely. Because the apicoplast's biochemistry is unique to the parasite and plants, drugs designed to inhibit its specific enzymes can kill the parasite with potentially fewer side effects on the human host. It's a major avenue for developing new antimalarial drugs, and even some herbicides work by targeting plastid pathways. Evolutionary history providing modern medical targets.
Incredible connections. Okay, so let's try and wrap this up. We started with a simple equation, but we've unpacked a complex engine.
We really have. We've seen the two distinct phases. The light reactions capturing solar energy in the thylakoids, making ATP and NADPH.
And the Calvin cycle using that energy in the stroma to fix atmospheric chiarose into sugar, fighting against the inefficiencies of its key enzyme, Rubisco.
And we've seen the amazing evolutionary adaptations C and seam that plants develop to overcome those inefficiencies, plus the intricate ways the whole process is regulated and integrated with the rest of the cell.
It really drives home the connection between this microscopic biochemical factory and the composition of our entire atmosphere and the foundation of life as we know it.
Absolutely. Which leads us to maybe a final thought for you, the listener, to ponder. We mentioned Rubisco's inefficiency, right? That it often binds oxygen.
Yeah, the photorespiration problem.
Because it's relatively slow and makes mistakes, plants have to compensate by producing absolutely massive amounts of it. In fact, Rubisco is often cited as the single most abundant protein on Earth by weight.
Just because plants need so much of it to get the job done.
Exactly. Think about the sheer amount of cellular energy, NCP, resources, nitrogen that a plant has to invest just to synthesize and maintain these huge quantities of this one, flawed but essential protein.
So here's the provocation. What if the synthetic biologists succeed? What if we could engineer and deploy crops with a significantly improved Rubisco? Perhaps one inspired by CO efficiency, one that strongly prefers Keo and barely ever binds O.
Consider the cascade. Less energy wasted on photorespiration means more energy for growth. Huge potential boosts in food production, especially when it's needed most. Less need for the plant to make quite so much of the enzyme itself, freeing up resources. And maybe, just maybe, a measurable increase in the rate at which the biosphere draws down atmospheric CO.
Improving this one, ancient, slightly broken biological light engine. Could it be one of the keys to tackling some of our biggest global challenges in food security and climate? Something to think about.